Hi students, in last class we have discussed about the use of microbes in industry. In last class we discussed about the general consideration and the production of amylase and several enzymes are produced from several microorganisms. Examples we are good. Then general consideration for the production of enzymes. The steps involved in the production of enzyme. First step is the selection of organism. Second step is the formulation of medium, and the third step is the production process, and the fourth step is the recovery and purification of enzymes. I already discussed the production of amylase by two techniques known as submerged fermentation and solid state fermentation. Today we discussing about the production of catalase. Okay, catalase is an enzyme that catalyzes the reaction by which hydrogen peroxide decomposes into water and oxygen. And this catalase is not only found in organisms leaving the presence of oxygen. And this catalase prevents the accumulation of peroxide, that prevents the damage of cellular organelles by the bad effects of peroxide. And it is used for preserving foodstuffs. And in the production of catalase, the catalase is generally produced from the fungus known as Aspergillus niger by the technique known as solid state fermentation process. So we have already discussed the solid state. What is solid state fermentation? So you should write the solid state fermentation, the production of catalase by using Aspergillus niger. Next is the production of peroxidase. The peroxidase is an enzyme that decomposes hydrogen peroxide by the oxidation of wide range of phenolic and non-phenolic substrates. The peroxidase are abundantly found in the bacteria, fungi, algae, plants and animals. And several microorganisms like bacteria, cyanobacteria, fungi, yeast are used for the production of peroxidase. And the peroxidase is used in decomposition of pollutants, production of animal feedstock and raw materials for chemical, agriculture, paper industries, textile dye degradation, paper pulp industry for lignin degradation, dye decolorization, sewage treatment, and also as a biosynthesis. So peroxide has a wide applications. Next is the production of lipase. Lipase is an enzyme that breaks down dietary fats into fatty acids and glycerol. So lipase converts the dietary fat into fatty acid and glycerol. The several types of bacteria and fungi are capable of producing lipase. A fungal species, when we are using the fungal species, we are using solid state fermentation technique for the production of lipase. When we are using bacterial species, we are using submerged fermentation for the production of lipase. So you should write the solid state fermentation by using fungal species, and you should write the submerged fermentation by using bacteria in the production of lipase. So already we have discussed the solid state fermentation and submerged fermentation in last class. So you should write the production of lipase by fungal species as solid state fermentation, and you should mention the solid state fermentation in the production of lipase by using fungus, and you should write the the submerged fermentation, it is lipase production by submerged fermentation plus bacteria in the production of lipase. Next is production of protease. Protease is an enzyme which hydrolyzes the peptide bonds joining the array of amino acids in the protein. Thus the protein is cleaved into amino acids. Proteases are produced from several bacteria like Bacillus, Pseudomonas, Clostridium, and also the proteases are produced from several fungi like Aspergillus niger, Aspergillus oryza. So you should write the production of protease, and the protease is produced from bacteria like Bacillus, Pseudomonas, Clostridium, protease, and protease is produced from fungus like Aspergillus niger, Aspergillus oryza. So you should write the production of protease. You should write the general consideration for the production of enzyme. Okay. The last enzyme is production of benzylase. Penicillin is a bacterial enzyme that inactivates most of the penicillins. So penicillins are capable of inactivating the penicillins, and it is an extracellular enzyme produced by the members of coliform group of bacteria by the Bacillus species and some of the strains of Staphylococcus. So penicillin is produced from different microorganisms like bacteria, Bacillus species and Staphylococcus species. And this penicillin is obtained from Bacillus subtilis, Bacillus cereus like microorganisms. And you should write when the question about penicillin is when you are write the general consideration for the production of penicillin. You should write in the penicillin's production, and you should specify the microorganism name, that is penicillin is obtained from Bacillus subtilis, Bacillus cereus, and industrially produced and exercise action same extent or removal of penicillin via specific inactivation. Okay, you should write the name of microorganism, that is important. So all are same, that is production process are same. As a general consideration, already we have discussed the production process same, but the different is that it's species, that is it's microorganisms. Okay, this is a industrial production of enzymes by using microorganism. I hope you are following my lecturing. Thank you.